In the present lecture we are going to study about various different types of polysaccharide complexes. These complexes are important for various physiological functions. The first one in this regard are called as glycosaminoglycan. Glycosaminoglycans are also called as mucopolysaccharides. They are present in the extracellular matrix. They are found in a variety of body structures including connective tissue. Along with several different types of proteins, glycosaminoglycans constitute the extracellular matrix. Glycosaminoglycans are unbranched polysaccharides. They have high molecular mass. They are made up of repeating disaccharide units. They have alternating uronic acid and hexosamine residues. Remember that uronic acids are oxidized derivatives of monosaccharides which contain carboxyl group on the last carbon. In this figure, you can see two common uronic acids, namely glucuronic acid and galacturonic acid. You can see here that they both contain a carboxyl group on their carbon number 6. The other type of monomer present is a hexosamine. These are the derivatives which contain an amino group on their carbon number 2. Here you can see two common hexosamines, namely glucosamine and galactosamine. In glycosaminoglycans, the uronic acid is linked to the hexosamine through a beta-glycosidic bond. On the next slide, you can see hyaluronic acid. It is the most common type of glycosaminoglycan. It is present in the ground substance, extracellular matrix, vitreous humor of the eye, and acts as a lubricant in joints. It is made up of repeating disaccharide unit which contains glucuronic acid linked to N-acetylglucosamine through beta-1-3 glycosidic bond. You can notice here that because of the presence of carboxyl groups, hyaluronic acid is negatively charged. Also, these carboxyl groups interact with cations. In this figure, you can see the structure of hyaluronic acid. The monosaccharide unit shown on the left is glucuronic acid. The carboxylate group is shown in red color. It is linked to N-acetylglucosamine. The N-actyl part is shown in blue color. The two disaccharides are joined through beta-1-3 glycosidic bond. Hyaluronic acid contains many negative charges on it because of the presence of carboxylate groups. Also, it contains many polar hydroxyl groups. Because of these polar groups, they are highly solvated and are bound by many water molecules. Also, these are high molecular mass molecules. The carboxylate groups are negatively charged and the repulsive forces between them result in making the molecules extended. Because of these properties, hyaluronic acid molecules occupy a large volume. So they act as a shock absorber. When pressure is exerted because of compression, the associated water molecules move away. But when pressure is removed, the polar group again take up water molecules due to solvation. Hence it acts as a shock absorber. Also, the molecules contain negatively charged groups which repel each other. So these molecules can come close to each other only to a certain extent. These extended molecules form parallel sheet-like structure where they slide over each other. This also contributes towards their function as lubricant. They are present in many different parts of the body and in joints as well, where they act as lubricant and shock absorber. An enzyme called as hyaluronidase is needed for the breakdown for hyaluronic acid. This enzyme is also present in snake venom and certain pathogenic bacteria which utilize it for disintegrating hyaluronic acid. This next slide shows the structure of several important types of glycosaminoglycans. You are required to remember the structure of only hyaluronic acid and heparin. The first one is hyaluronic acid. Some other important glycosaminoglycans include chondroitin sulfates, dermatin sulfate, and keratin sulfate. As you can see, glycosaminoglycans contain carboxyl groups as well as sulfate groups and hence are extensively negatively charged. So, by means of these negative charges and the polar hydroxyl groups present on them, these glycosaminoglycans act as a lubricant and shock absorber. The last one shown in this figure is heparin. Heparin is a naturally present anticoagulant in blood. Anticoagulant are those substances which prevent the process of blood clotting, whereas clotting factors carry out the process of blood clotting. Both clotting factors and anticoagulant are present in blood. An equilibrium between these exists in blood and maintains blood in its physiological fluid form. Heparin prevents the formation of blood clots in the blood vessels. Warfarin is a synthetic anticoagulant. It is used as a rodenticide. When rodents consume this anticoagulant, they suffer from excessive internal bleeding and therefore die from blood loss. The next type of complexes are called as proteoglycan. Proteoglycans have several important functions. They act as organizers of tissue morphology. They act as a filter and regulate the transport of substances. They are also important for cell signaling. As the name suggests, proteoglycans are made up of a core protein to which glycosaminoglycans are attached. The table on the next slide shows various important proteoglycans. This table is for your information only. You are not required to remember all these names. This next figure represents the proteoglycan structure of an integral membrane protein. Integral membrane proteins are those which pass through the membrane as shown in this figure. On the outside of this cell, you can see that different types of glycosaminoglycans attached to this integral membrane protein. On the outside of this cell, you can see that different types of glycosaminoglycans attached to this integral membrane protein. On the next slide, you can see a complex assembly of proteoglycan. In this next figure, you are looking at a cross-section of the cell membrane. This is the inside of the cell where you can see actin filament, which are cytoskeletal proteins. This is the inside of the cell where you can see actin filament, which are cytoskeletal proteins. These cytoskeletal proteins are attached to the integral membrane proteins as shown here. And these membrane proteins, in turn, are attached to the proteoglycans on the outside of the cell. Interactions of cells with proteoglycans are important for tissue modeling and organization of cells. Another important complex of carbohydrates with proteins is called as glycoprotein. Glycoprotein are those proteins which contain carbohydrates covalently attached to them. This process of attaching carbohydrates to these proteins is called as protein glycosylation. The process takes place when the proteins move through the endoplasmic reticulum Golgi complex pathway followed by protein synthesis. Glycosylation of protein is important for their physiological function. For example, antibodies produced by B lymphocytes of the immune system glycoprotein. Several other glycoproteins are needed for other cellular functions. Oligosaccharides are attached to proteins through two types of chemical groups. In the figure on the left side, you can see O-linked glycoprotein. These are named so because the oligosaccharide is attached to the protein by means of an oxygen atom. This linkage is mediated by those amino acids which contain a hydroxyl group on their side chains, mostly serine or threonine. The other type of linkage is called as N-linked glycoproteins because this glycosylation takes place by means of a nitrogen atom. This is shown on the right side of the figure. The amino acid asparagine is required for N-linked glycosylation as shown here. Different oligosaccharides attached to these glycoproteins are shown by means of these symbols below. The next type of carbohydrate complexes are lipopolysaccharides. As the name says, this is a complex of carbohydrates with lipids. Lipopolysaccharide are found in the membrane of gram-negative bacteria. This figure shows the structure of lipopolysaccharide. These are the fatty acyl chains corresponding to the lipid part of the molecule. And here the carbohydrate part of the molecule is shown. Lipopolysaccharides are important as recognition marker and antigens. Carbohydrates present on the surface of the cell are also important for cell-to-cell introduction. There are several different types of carbohydrate binding proteins present on the cell surface. One such type of binding proteins are called as lectins. This figure shows the process of leukocyte extravasation. Leukocytes float freely in blood. When there is an infection, inflammation in tissues, the leukocytes bind to selectins on the blood vessel endothelium and start rolling. Eventually they squeeze themselves and most through the spaces between cells and reach the site of infection, injury in tissue. This process of movement of leukocytes from blood vessel to the tissue is called as extravasation. In this next slide, you can see a microscopy video of an actual capillary. You can clearly see a leukocyte rolling along the capillary wall. This rolling is mediated by specific carbohydrates present on cell surface. The leukocytes eventually squeeze themselves through the inner capillary wall and migrate to neighboring affected tissue. These cells are shown by 3 red circles below the capillary. The next slide shows interaction of different cell types by means of carbohydrate binding proteins. You can see that different cells of the body, for example lymphocytes shown here, bind to specific binding protein on other cells. Bacteria and viruses also have specific carbohydrate binding protein on them. They attach to host cell by means of these carbohydrate binding proteins. In this next micrograph you can see many elongated bacterial cells attached to host epithelial cells. The bacterial cells shown here are E. coli cells. They have a protein called mannose binding protein. This protein binds to the mannose residues on the surface of host epithelial cells. This attachment is an initial step to colonize and infect host cells.